No, please keep asking questions. It's good for the recording to have all these questions asked. It's a good thing. More people can ask questions aside from Hannah. Yeah. You're just going to be listening to your own voice on these recordings. It's going to be great. Just Hannah. Phil's helping too. Okay, so when part of your brain tissue dies, which is the end result of this, right? You got conked on the head, you had an initial insult, you had a coup contre coup injury, you had ischemia, you had maybe a hematoma form, maybe it was a subarachnoid hematoma, I don't know. Maybe you had inflammatory events, maybe you had a reperfusion injury. Some combination of everything we've covered so far has happened, and now some of your cells have died. In your brain, you're very likely to develop liquefactive necrosis, and that's just going to sit there. This little jello textured mess is going to sit there. So an abscess may form in your brain. If it's infectious, then that's going to be bacteria-based, but not necessarily. This is really just demonstrating liquefactive necrosis right here. But you could also have a localized collection of pus as well. If it's bacterial, then you'll need IV antibiotic therapy to deal with that, or possibly drainage or excision of the abscess. Oh, we never did watch that hematoma video. I'm going to pause so we can watch the hematoma video before we move on to the rest of these. Questions we asked while watching that video was, do you put that bone flat back on after that removal? And as my understanding is usually no, you would not put that bone flat back on. You can improve perfusion pressure by relieving that pressure on the brain. If there's an increase in intracranial pressure on there, you're not going to put that bone flat back on. Okay, so more physical brain disorders. This one's going to get a little bit more neurological, though, than just literally getting conked on the head. All we've addressed so far is getting conked on the head, right? Now we're going to move into seizure disorder. We're not just going to say flat epilepsy. We've already seen a condition other than epilepsy that leads to seizures, right? There's more than one thing that leads to seizures than just epilepsy. Forms of dementia, we'll talk about Alzheimer's and vascular dementia, and we'll also be talking about some of the competing hypotheses about Alzheimer's dementia because it's not really that clear-cut. Then we'll move into Parkinson's, cerebral palsy, hydrocephalus, and cerebellar disorders. So with seizure disorders, anything, and again, all a seizure is runaway synchronicity of neurons. Neurons are firing without their normal input, and they're firing in a way that is synchronized. That's not supposed to be synchronized. Instead of having one circuit fire that controls your arm movement, that circuit and everything regional to that is also firing without the correct inputs. And why that happens is poorly understood. At some level, it has to be some kind of electrical disturbance. It could be an ion disturbance, but again, we haven't tracked down that ion disturbance. We haven't said, oh yes, it's sodium. We have not found that. So the manifestation is going to be related to, again, the territory that is being activated. It could be very, very well localized. If it's a response to a very specific AV malformation, for example, it would be localized to just that area. The guy I knew with the AVM, his was very frontal lobe, right? So his manifestations would be very related to that spot on his frontal lobe. Whereas it's also possible to have very global seizures that impact large portions of the brain. So those are going to appear differently. So epilepsy or seizure disorder specifically refers to recurrent seizures. This would be like the primary disorder, would be epilepsy. Whereas AVM at the seizure would be secondary. So a cerebral injury, a lesion, a metabolic or nutritional disorder, or idiopathic. Sometimes we have no idea why somebody is epileptic or has seizure disorder. We could say, okay, these neurons for some reason are abnormally sensitive to change. Again, why? We don't really know. Do they have more sodium channels? Do they have more AMPA receptors? Do they have more this receptor, that receptor? We don't know. But either way, they're going to be abnormally active. A person with a seizure disorder may experience an aura or a prodrome period before a seizure. They may be aware that they are about to have a seizure. May or may not be able to take steps to keep themselves safe during that seizure. So we have our stereotypical seizure. Somebody drops to the floor and starts shaking. That's only one form of seizure. That's a tonic-clonic seizure, classically known as a grand mal seizure. And that's, again, that's the stereotype. They do exist, but that's just one kind. More common would be just an absence seizure. And this is more likely to occur in small children. This is where your kid spaces out and you can't get their attention for anything, and then they come back to you and they have no idea that that time passed, that you were trying to get their attention. They're simply not there. So remember, some of our neural circuits are excitatory, some of our neural circuits are inhibitory. So runaway synchronicity could mean everything stops if it's that circuit. Atypical absence, and the staring spell goes with some myoclonic jerks, which is the shaking. There's myoclonic, which is single or several jerks. It might be regional to one specific body region. The atonic seizure is a fall down. It's a drop attack that may not be accompanied by any kind of shaking. It would just be a fall. And then we get back to our tonic-clonic or grand mal seizures. So again, it's just about how much of the brain was involved and what regions of the brain were involved. Yes?
Are tonic-clonic, are they the most common?
As I understand it, no.
Okay.
Yes.
So I'm going to get with epilepsy monitoring, and sometimes we'll get patients that are having your typical tonic-clonic grand mal, but we EEG them and there's no like evidence of seizure activity. Like, I just don't understand how someone could not necessarily, well, I guess fake that for lack of better words.
Yeah, I don't know.
It's like they make it seem like it's actually real, like they're having all the manifestations.
Right. So are these people with established seizure disorder, or are they coming in, like, where are they coming from?
They're coming in to like figure out why they're
having a seizure. Okay. Were they usually referred there? Are they there by their own will?
Yeah, they come in like for like a program, like they'll be there for like a whole week.
Gotcha. I don't know. It could be that like you think they're faking it. There's some neurological issues. Do you have some insight into that? Does somebody else have insight into that? Or do you just have more questions? I want to try to finish answering this one before I move on. And again, I don't know that I have an answer for you. There are mental health conditions where people are seeking attention. Munchausen syndrome, for example, or hypochondriacs. There could be muscle spasmings that are unrelated. Yeah.
Well, I suffered seizures when I was about 17 for about like 6 months, and it was because I was given the Depo shot as a form of birth control, and what it did was it caused me to have such horrific seizures that it was like, or migraines, that it was causing my body to like express the pain in a seizure way. It was the craziest thing, and I couldn't do anything about it. And yeah, and I was like, I mean, and so and they tried EEGs and they were like,
and there wasn't anything.
There's nothing going on in my brain, but I mean, like.
So the migraine and the pain was what was causing sort of jerking muscles.
Like a body, like it was just my body reacting to that pain. It was so insanely bad.
So you had a question?
I read this study about how some people can have behavioral overload seizures, like when they're so overwhelmed that their body, instead of having like it's kind of like an advanced panic attack to make their body relax. We saw with this girl where we put her in CT, and she had an atopic seizure, and her eyes started moving, and her feet were twisting, but that's it.
Yeah.
She responded to like ocular reflex, like we squirted the water in her ear, and she responded to that, but she couldn't stop her eyes from moving, and she couldn't respond to it.
So people can have seizure-like activity without it being a sign of electrical activity in the brain, is what we're saying. Yeah, I mean, we know about tetanic contractions, we know about control, we know about muscle twitching and cramping. As for why Depo-Provera would cause that, I mean, it's some.
It was a reaction to the migraine.
Yeah, so the migraines as a result of that. Yeah. Blood pressure changes with birth control are very dangerous for a lot of people. Yeah. What's that? Febrile seizures. Let's come back to that concept. So somebody's feverish and having a seizure as a result of that. Again, this part, what's that? Okay.
And she says it's not how high the temperature goes, it was how fast the change.
Okay.
So, like,
Yeah. So abrupt temperature changes.
So, yeah, abrupt temperature changes in children.
That's cool.
They don't have that mechanism to stabilize it, causes the febrile seizures. So she said, I don't care if they hit 106. Now, if they went from 99 to 106 in an hour, I can.
Okay.
So she's like, it's more of how quick it went. So for children that were very sensitive to those changes, at the first sign of being sick, it was Tylenol around the clock.
Yeah.
Just
that makes sense.
It. And after like 7 or 8 years old, things happen.
Yeah. And so that in a way does all track. None of that would be epilepsy, right? But all of those seizures, or most of those seizures, would be caused to changes in the neurological environment, right? It's environmental. It's not a specific circuit firing. Okay, now some people with epilepsy are going to have their seizure and then recover, have a period of without seizure. Some people are going to have just ongoing seizures. Status epilepticus, continuing series of seizures without a period of recovery. Of course, that's going to be the most dangerous because when do you eat? When do you clean yourself? When do you do literally any activity of living? Yeah. And again, there's going to be changes. There's going to be difference between a very localized seizure or partial seizure versus a global, a complex seizure. So you can start at a partial seizure and move global or not. It's scale independent, much like the heart issues were. Okay, different kinds of dementia. And we'll just be differentiating. That was awesome conversation about seizure, by the way. I don't think any cohorts had that much contribution to the seizure discussion before. Usually we just ask about it and move on. Yeah.
I've got one seizure.
Sure, go for it.
So I have a nephew, and he has epilepsy, and his mom basically explained to me that, like, after he has seizures, like, it sets his brain development back. So he's, like, 6 and acts like he's 2.
Yeah.
Is that, like, normal? What's happening to the
That
brain?
makes sense. So the brain is very plastic when you're very, very young. And we have this saying in neuroscience that neurons that fire together wire together. So part of your neurological development, you're born with all of your neurons. They're already there, right? Because they're non-mitotic in the adult. So you're already born with them. The process of development and growing up is actually a process of synaptic pruning. So there's actually one hypothesis. This gets super interesting. I'm glad you brought that up. One of the hypotheses is that we're all born with synesthesia, which is the condition where, like, you can smell colors and hear shapes, and so your senses are all cross-wired to start with. And again, this is a hypothesis. We can't exactly ask a baby if they can smell colors, right? But we think that, when you touch this thing, then that sensory pathway fires, and then it goes back to the correct part of your brain, and that fires together, so it wires together. And then the thing that is not that will decay. You'll prune those synapses that are the incorrect connection. But when your neurons are globally firing, then it's going to keep those synapses that are incorrect, right? So that's what I'm imagining is happening with the delayed development is you're not getting the correct pathways forming. You're not getting the correct synaptic pruning. And so they're going to, behave as somebody who hasn't been there. There could also be some other metabolic changes that are going to potentially even cause brain damage or be a response to brain damage. It's going to be a circuit of causing brain damage, being brain damage, resulting in brain damage. So none of it's good. No, we need more treatments for epilepsy. And there are some promising treatments for epilepsy in the pipeline. Some of them are currently illegal to research.
It's been really responsive to CBD. I live in Washington. So.
Yeah, I was debating whether I was going to bring up CBD, but you brought up CBD, so I can bring up CBD now.
Multiple hours to going weeks without.
Yeah.
It's amazing. We took them off Keppra and all the other drugs, and now they're just on CBD. And you see so many reactions are different. And Klonopins too. And they're doing so much better. It's amazing to see. I think 3 out of the 4 of the kiddos I take care of right
Yeah, there's a lot of citizen science going on with CBD because it's still technically regulated. Colorado did just legalize hemp, and there are a lot of national movements towards legalizing hemp. It's still technically Schedule 1, even though it is not psychoactive. CBD itself is non-psychoactive. It will not get you high. It will not get your kids high. It will not get anybody high, just the CBD. Yeah, it's a big problem. Go for it, and then Phil.
Can he start to develop at a more normal rate? Will he like advance in age and it's not permanent?
It depends on, at 6 years of age, there's still some brain plasticity. The issue is critical periods for certain things like language acquisition. So if he's still stuck at age 2 language acquisition, that may not develop at the same rate, but certain skill sets, if the seizures stop, the brain is still very plastic at the age of 6. There's, I think, a famous case of a young girl who had a hemispherectomy, entire hemisphere removed at the age of 5, I want to say, and she's got some hemiplegia, not quite. She can walk. Yeah, so there's a young girl, age 5, hemispherectomy, and I think it was to treat epilepsy as well. And she had literally one hemisphere removed at the age of 5. And she was shot. There may be more than one person with this though. So she's walking around. She can play. She can talk. She can go to school. She can do everything. So again, the young brain is very plastic. And again, even if you don't replace those neurons, there's a lot more compensating that can happen at that age. There are a lot more stem cells around, so there can be more healing. So the sooner the better is the rule there. With CBD, again, it's non-psychoactive. The mechanism of action is poorly understood. It's acting on the endocannabinoid system, which we've more recently identified as mainly a pain-killing system. We've actually decided that runner's high is more likely to be endocannabinoid rather than we used to think it was dopamine rewards pathway. It's probably not. So when you have a runner's high, it's probably a little bit more closely related to like the way THC makes people feel. But it actually seems to be, by some sources, a partial antagonist to THC. There's some evidence that it's a partial THC antagonist. It actually acts against that. So if, for example, THC is making somebody feel paranoid, the CBD actually takes away that anxiety and paranoia. So there are a lot of health claims around CBD that we can't ethically make yet. But like I said, there is a lot of citizen science showing that very high doses, around 300 milligrams of CBD, is showing some effect. It's citizen science, and we have to take it as such, and we can't say these through the FDA yet. So we're not going to prescribe it yet. It is a big deal. Right. Oh, no, yeah. I mean, the CBD is a big deal. Like, it's such a big thing. It's going to fix so many lives. It's all about the money. Yeah, Phil's got it. It's all about the money.
Just a skosh of meth and your seizures will stop. Then I'd be like, whoa. Yeah.
Sure. I'll post some neuroscience worksheets for you. Absolutely. And if you guys want to help me figure out which worksheets are most useful to post from neuro, we'll do that. Okay, that was awesome discussion on seizure. And again, the CBD is controversial. It's still technically illegal, so I can't advocate it. My hands are tied. Okay, Alzheimer's, dementia, or vascular dementia are the two major kinds of dementia. Now, vascular is really straightforward. It just means you had a cerebrovascular event, maybe an aneurysm burst and there was a hemorrhagic stroke, maybe there was an ischemic stroke. Part of your brain died. That's vascular dementia. And the part of your brain that died was related to memory. Alzheimer's dementia is really poorly understood. And I was just having this conversation with Enrique a little bit earlier that there are competing hypotheses for what Alzheimer's dementia actually is. And again, lots of people have written papers on Alzheimer's dementia, and sometimes they get to one hypothesis, sometimes get to the other hypothesis. They never really get to all the hypotheses. It's a hard thing to address in one paper as well. So with Alzheimer's disease, what we do know is that neurons are degenerating, especially in the hippocampus. They're going to say temporal and frontal lobes. That hippocampus is in the temporal lobe. There can be some general brain atrophy. We do see this thing called beta amyloid plaques. So beta amyloids, I may have mentioned them before because they're related to sleep. So beta amyloid plaques are like tau proteins. They are a metabolic byproduct. Your brain metabolism does things normally. It produces beta amyloids. They get removed to the space between your neurons. They get to your extracellular matrix, right? But they are hard to clear out. So while you sleep, you are clearing out your beta amyloid plaques. That's one of the reasons that they're important to me, is that sleep may be preventing Alzheimer's. That's one hypothesis. It's not a perfect one, though, because plenty of people with sleep disorders do not develop Alzheimer's disease.
I also read a study that they said India had a way less rate of Alzheimer's, but they were relating it to turmeric and saying that turmeric helped clear the plaque off of your brain.
Turmeric's a blood thinner, so it's possible. It's an anti-inflammatory and it's a blood thinner. So I don't know.
Like, I was just going to say what they were relating to.
I mean, turmeric's good stuff nutritionally. Yeah, not gonna lie. It is possible to OD on turmeric.
Yeah,
You can hemorrhage. Yeah.
So just double check it before I said it out loud. But yeah, in January there was an article published about periodontal disease bacteria related to.
I'm about to get to that.
I was like, I'm like, wait.
I was at a seminar at the anatomical conference, so we're about to talk about it. Okay, so there have been investigations into the genetic cause. Why are we building up beta amyloid plaques? Why do we have neurofibrillary tangles? Why do we have brain atrophy? Is it purely genetic, is one thinking process. Another more recent one that I was about to bring up, which is exactly what Jessica said, is that it could be infectious inflammatory. So when I was at this guy's thing, and he was kind of burying the lead a little bit, it wasn't even called like periodontal disease leads to Alzheimer's. It was infectious, inflammatory, blah, blah, blah. So what he did basically, or what they did, this team, is they were investigating sort of the evidence of antibodies and looking for those antibodies and where they were. And they did find antibodies against the specific bacteria associated with periodontal disease and gingivitis. They found those antibodies in those brain cells in those areas that were atrophied. So it could be that you had very poor oral health. You developed an infectious inflammatory disease that infiltrated your brain, you developed antibodies to it, and you caused some damage in your head. Yeah, it impacts your heart, of course. You've got microvasculature, and that's a big deal, Ellen. That one's controversial too, though. Yeah. Right.
They said if you have metal
I'm gonna go with a no on that one. Yeah, no, don't. I agree. Don't shoot the messenger. I have a lot of hippie friends who are doing like, oh, I got all of my mercury fillings out, and now I feel so much better. Yeah, we used to fill fillings with like mercury and some heavy metals and things, and some people think it's leaching, and it could be. I'm not gonna, but I've never seen any evidence in favor of that being a thing, especially on the cell phone use. Cell phone use is not in evidence for anything. It correlates with a lot of things, but a lot of things correlate with a lot of things. Cell phone use correlates with autism. You know what? Organic food correlates with autism diagnoses. So is organic food causing autism? I don't know. Could be. So correlation is not causation. There are all kinds of things. And I think the recommendation is generally, if your feelings aren't bothering you, don't do anything to them. And even with, have you guys heard about the breast implants that cause breast cancer? So there's textured breast implants specifically actually harbor certain bacteria is what we're finding out, and that can irritate the breast tissue and it can mutate and you can develop breast cancer. They're still not recommending that women have them removed unless it's bothering them.
Have you heard of like keeping your cell phone in your bra gives you breast cancer? My mom thinks
A lot of people think cell phones are causing cancer. I'm not saying it's impossible. You never know. I mean, we used to like put uranium in beauty products. Humans are really bad about just developing technology and running with it until they find out it causes cancer. But there's no evidence for it right now. Okay, so there's the infectious inflammatory model, there's the genetic model, there's an interaction between genetics and infectious inflammatory. There's yet another competing hypothesis that Alzheimer's dementia is just a form of vascular dementia where the vascular insult is non-detectable. Like maybe you had just a series of brain bruises and TBIs and really small things that led to non-detectable vascular issues that nevertheless manifested as Alzheimer's disease. So one more fun fact before we get to your next statement, because it looks like you want to say something, is, I forget what I was going to say. Oh yeah, in any Alzheimer's diagnosis while a person is alive is provisional. We're not diagnosing Alzheimer's for real in living people. We're saying you have a form of dementia, we do not see a cerebrovascular insult history, therefore we're going to call it Alzheimer's. We don't know it's Alzheimer's until they're dead and we cut into their brain and we look at their hippocampus. Was that your question?
No, I was going to say the CT, they were also, it presents a lot like Alzheimer's,
Yeah.
like not remembering things and like severe agitation and all this crazy stuff, and like
Yeah.
the CT, you can't diagnose it until they're dead because, and their brains look a lot like the brain of like Alzheimer's.
Exactly.
But like way too young to have the brains that they did.
Mm-hmm. Absolutely. So yeah, the takeaway is for Alzheimer's dementia, we don't know. We're not sure. A lot of people are doing research on it. It's a hot button issue because our identities are our cores of ourselves, and having our identities removed from us is one of the worst things that humans can imagine for themselves. So the research is there and it's interesting, and that infectious inflammatory model is really cool. It means take care of your teeth. The end, right? And that's huge in its own way. Just take care of your teeth and you'll be fine. So we do mostly talk about memory loss. We'll start with sort of minor memory loss. We'll progress to more severe memory loss. And again, there's more areas of research. It's more complex than I'm letting on and saying, some parts of the brain may be more impacted than others. Maybe you'll remember lyrics to your songs, but maybe you won't remember your life events. And there's all manner of research about what goes and what stays. It's not just about memory. There's going to be progression that may include problems with problem solving, communication, activities of daily living. We may start losing some of our autonomic stuff, eating, swallowing, bladder and bowel control. Again, as more parts of the brain degrade, and that's just regional. We think of dementia in terms of the hippocampus, but ultimately, if you go back to that severe Alzheimer's brain, yeah, our hippocampus is pretty gone. There's the hippocampus, there's the hippocampus. But you can also see there's general wasting that's occurred in the long term. Okie dokie, yes. So that's your line for hospice care for Alzheimer's?
So when you finally get to that point, they can bill under dementia or vascular dementia. They can't bill under Alzheimer's for obvious reasons.
Yeah.
But when you get to the point where you're starting to have bowel or bladder incontinence, or you're starting to try to wander outside of your house, you're at the point where you're now eligible for hospice care.
Gotcha.
Because we know that you now have about 6 months.
Gotcha. Okie dokie. Let's move on to Parkinson's disease. This is going to require that you remember something related to motor activity in the brain. So what are the 3 motor regions or motor areas of your brain? What part of your brain still? So precentral gyrus is one. That's going to initiate motor movement. Well done. What else? 2 other motor things that do motor, other aspects of motor, other than initiating that movement. Cerebellum, that's going to be fine-tuned motor movement. It's going to allow that hawk to predict the movement of that rabbit and grab it while it's flying, which is really complicated. It's going to enable you to learn how to play the piano. Cool, right? What's the third and the one we're looking for right now for Parkinson's? Nope, hippocampus is memory. Nope. It's the most commonly forgotten. This happens every time. It's not just you. Basal ganglia, ding, 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 ding. Also known as cerebral nuclei. The AMP 1 and 2 textbook refer to them as cerebral nuclei. We're going to call them basal ganglia in your patho textbook. Basal ganglia is a bit of a misnomer. That's why they changed the name. Usually ganglia exist outside of the central nervous system, but they are basal. So their cerebral nuclei are basal ganglia. Awesome. So what do the basal ganglia do? Nope. That's the colliculi, which we'll need those in a bit too. I'm glad you remember the bus story. It's an important one. Okay, what do the basal ganglia do? Initiating and controlling, kind of, yeah, they're going to be related to initiating and controlling motor movements, which is why it's going to tie into Parkinson's. There's a specific way that I phrased it when I taught it to you guys. There's two different pathways in the basal ganglia, the direct and indirect pathway, which are actually pharmacologically about to be very important to you. That's a reflex. Nope. That doesn't even need to go to your brain, so no. But again, you still need to understand those reflexes, because that's going to come into play too. Nope.
Oh.
Do you want me to pause while you guys figure this out? Pausing. Sort of got to it. We got to that in your basal ganglia, there's two competing pathways. There's an excitatory pathway and there's an inhibitory pathway. And what this gets, and the phrase that I was looking for, is not too much or not too little movement. Does that just simply ring a bell? Okay. All right. Not too much, not too little. So what we mean is that, yeah, we've got our motor movement initiation, right? But there's also going to be signals going on all the time that make sure that you're not moving when you don't want to be moving and that you can start moving when you want to start moving. And Parkinson's disease is a disorder of both. So, now notice up here, the first thing that we're talking about for manifestation of Parkinson's disease is a general lack of movement. You associate it with the involuntary shaking. But somebody with Parkinson's is also going to have a flat affect, which means like a blank mask-like face. They may have a hard time initiating the movements they want to make, while simultaneously making movements they do not want to make. So that's why I phrased the basal ganglia back in the day as two just competing pathways that cause not too much, not too little, right? That's how you want to think of those basal ganglia. And they're going to interact. They do project directly to that primary motor cortex on the precentral gyrus, and then that sends that out via the corticospinal pathway to cause your movements, right? All of that circuitry, that is going to, understanding that helps you understand our treatment protocols for this, because they get a little wacky. Now, the pathway to those basal ganglia, that's in the substantia nigra of the midbrain. You had to learn that in AMP 1 as well. Substantia nigra of the midbrain, that black stuff, which is literally what that translates to, is the metabolic byproduct of producing dopamine. So the indirect and direct pathway of the basal ganglia, those are dopaminergic pathways, which is why this is about to play in big time into pharmacology. When we mess with dopamine, we're going to mess with movement and emotions. Are you starting to see where this is going? Yeah, Parkinsonian tremor for certain dopaminergic drugs, right? Or dopamine blocking drugs, depending. Okay, and so that also means this is coming up next week when we talk about schizophrenia. Yes.
How do the deep brain stimulators work that they get for Parkinson's?
It depends. So we would be stimulating excitatory pathways, and then we would be inhibiting the excitatory pathways. So like we'd be doing the opposite. I don't know if I said that the correct way, but I think you know what I mean. Like if it's not working enough, we would stimulate it. For the involuntary movement, as I understand it, sometimes we ablate those motor pathways to get them to stop moving, which means they'll not have the voluntary aspect of it either. But that may be preferable to the involuntary movement. So ablative surgical procedures may be helpful for motor symptoms. There's no cure. We can temporarily increase the production of dopamine levels. That was L-Dopa, that was Patch Adams, and the laughter and the joy. But usually as we attempt to increase production of dopamine or replace dopamine, it's still, those levels are still going to fall. It's just going to slow it down a little bit.
Would that have, like what we were talking about last week, where if you, would your body stop producing as much if you were giving it exogenously?
So there's downregulation, and then literally what you can see in this brain, and I've seen this in real brain dissections, that substantia nigra, you just stop producing dopamine. So if the drug you're taking is to make you start producing dopamine, to give you dopamine precursors that you transform into dopamine, your brain is no longer capable of that. So precursor molecules won't help. Dopamine replacement might, but I don't know that we have those drugs. Ask Ashley if we have those drugs yet. And even then, you would see long-term downregulation of receptors. Fun times. So dopamine, yeah, it's a complicated thing. It's your rewards pathway. It's your motor pathway. And we're also going to see it in learning. So dopamine does a lot of things. It's about time for another break, though. It's 10:57.